Good morning. Today we're going to talk about bioenzymes. During this talk, you will learn what they are, their advantages related to other catalysts, and their industrial applications. Microorganisms have been used for thousands of years in order to make alcoholic beverages, bread, and fermented dairy products. These organisms are used as a fruitful source of enzymes, the most of which, nowadays, are isolated before use. Those enzymes, produced by living organisms, are called bioenzymes, from the Greek bios, which means life. Bioenzymes are used in several different fields, such as in industry, agriculture, and medicine. They come in handy for different reasons. First of all, an enzyme is a protein that, binding to a specific substrate, is able to catalyze a particular chemical reaction. Moreover, enzymes can operate at NTP, which is normal temperature and pressure, room temperature, atmospheric pressure of 1 atmosphere, and within normal pH ranges, usually around 7, thus reducing the production cost and being more efficient. Also, producing enzymes from living organisms is cheaper and faster than synthesizing the whole molecule in the lab. Eventually, these enzymes are biodegradable, so the waste disposal is simple and less noxious for the environment. Many industries rely on the large-scale production of bioenzymes. Commercial enzymes are produced from 4 main sources: plants, animals, fungi, and bacteria. Most enzymes used in industrial processes today are produced by microorganisms grown in bioreactors or fermenters. These enzymes can be extracellular or intracellular, whose isolation is more complex. Extracellular enzymes are secreted by the cell, while intracellular enzymes work within the inside. In both cases, microorganisms are grown in a sterile fermenter rich in nutrients. Here's a fermenter. Its conditions are closely monitored in order to optimize the growth of the microorganisms. When the enzymes have accumulated, the harvesting process begins and a powder of enzymes is produced through several different steps. So, bioenzymes can be produced from several different sources. However, industry takes advantage of just a few of them. In particular, the most used microorganisms are bacteria, yeasts, and fungi. The most common bacterium is Escherichia coli. E. coli can live on a wide variety of substrates, yielding different products. In addition, the bacterium is easy to engineer and it has a very short replication time. As yeast, Saccharomyces cerevisiae, commonly known as baker's yeast, is largely used. Historically, it has been employed in beer and wine making, and today it is behind almost every sort of fermentation. The most famous fungus is Aspergillus niger. The microorganism is largely used in industry because of the variety of enzymes it can produce, such as citric acid and pectinases.
Now we are going to present some industrial application of bioenzymes, such as the production of gluten-free and lactose-free products, dairy products, soft-centered chocolate, and fruit juice.
Gluten is a general name for proteins found in wheat, rye, barley. Gluten comes from the Latin gluten, which means glue. In fact, it helps foods maintain their shape, holding them together. Gluten also gives elasticity to dough, and it provides a chewy texture to the final product. Celiac disease affects approximately from 1 to 2% of the general population, but most cases remain unrecognized and undiagnosed, thus being a risk of serious, long-term health complications. Patients may suffer severe disease symptoms. Untreated celiac disease may cause malabsorption, iron deficiency, osteoporosis, and greater mortality. People affected by celiac disease must follow a very strict diet based on gluten-free products. The expression gluten-free is generally used to suggest a harmless level of gluten rather than a complete absence of the protein itself, impossible to ensure. On the other hand, there are many foods made from grain that do not contain harmful gluten. such as corn and rice in all forms, flours made from gluten-free grains, nuts, beans, and coconut. There are also totally gluten-free products such as milk, butter, plain yogurt, potatoes, vinegar, eggs. Because of celiac disease, in the last few years, new methods to produce gluten-free products have arisen. In particular, proteases are the most largely used enzymes in order to catalyze the reaction breaking down the gluten. Proteases are a group of enzymes that catalyze the hydrolysis of proteins. Proteases are produced naturally by many microbes, such as bacteria and fungi.
Lactose is a disaccharide derived from the condensation of galactose and glucose. It is contained in milk and dairy products. It is the major source of calcium for our organism that prevents diseases related to weak bones, such as osteoporosis. Lactase comes from the Latin lac, that means milk. Lactase is the enzyme produced in the epithelial cells of the duodenum, in the intestine. It splits lactose into the two monomers glucose and galactose, then easily digested. Some people's body do not make enough lactase, so they are not able to digest milk well. These people suffer from lactase deficiency, and they are called lactose intolerant. Lactose intolerance usually develops during the teen or adult years, when our body reduces the amount of lactase. Symptoms of lactase intolerance can be mild to severe, depending on how much lactase our body makes. They usually be in 30 minutes to 2 hours after eating or drinking milk products. These symptoms may be abdominal pain, cramps, diarrhea, and flatulence. The intolerance can be diagnosed with the hydrogen breath test, in which about 25 grams of lactose are swallowed by the patient. If the hydrogen levels in the patient's breath are high, he probably suffers from lactose intolerance. Another more invasive way to diagnose the intolerance is the intestinal biopsy, through which the levels of lactase in the patient's body is measured. Nowadays, the number of people who suffers from lactase deficiency is increasing, so this has stimulated the production of lactose-free products by the food industry. These products are not totally lactose-free. In fact, they just have a very little percentage of it, not yet broken down. These products rely on the production of the enzyme lactase that can be extracted both from yeasts and fungi. The enzyme is added sometime during the production process. People who suffer from lactose intolerance can take lactase tablets, pills containing the enzyme, in order to be able to digest foods with a standard level of lactose.
Chymosin, or rennin, is an enzyme of the family of proteases found in rennet. It's a complex of enzymes produced in stomachs of newborn ruminant mammals. It helps them curdle the milk they ingest, thus provide a better absorption. The word chymosin comes from the Greek chumos, which means juice. Rennin comes from the Old English verb that means to coagulate. As this verb may suggest, the enzyme has always been used in cheese making, thanks to its clotting property. Before the advent of genetic engineering, the enzyme was extracted from cows. Yet, this method would be too expensive and unsustainable. So, bacteria, fungi, and yeasts are engineered with rennet producing genes from ruminants. This way, these microorganisms become able to synthesize chymosin, which is then easily extracted. The most common microorganisms used in food industry are Aspergillus niger, E. coli, and Cloveromyces lactase. Rennin was the first artificially produced enzyme to be registered and allowed by the U.S. Food and Drug Administration.
The production of soft-centered chocolate relies on the liquefaction of the sucrose present in chocolate. This reaction is catalyzed by an enzyme invertase. Sucrose, commonly known as table sugar, is a disaccharide composed of glucose and fructose. This mixture of monosaccharide is called inversugar. Sucrose can be hydrolyzed in the presence of invertase. Sucrose can be hydrolyzed relatively easily. In fact, the reaction proceeds in an acid environment with the aid of invertase. However, in food industry, invertase is used instead of strong acid, such as sulfuric acid, which are noxious for us. Fructose is preferred over sucrose because it is sweeter and does not crystallize as easily. However, the use of invertase is rather limited because another enzyme, glucose isomerase, can be used to convert glucose to fructose more inexpensively. For health and taste reasons, its use in food industries requires invertase to be purified. Contrary to the most other enzymes, invertase exhibits relatively high activity over a broad range of pH. Commercially, it is mostly produced by yeast or fungi.
Pectin is a structural polysaccharide contained in cell walls of plants. It was first isolated and described in 1825 by Henri Branconneau. Pectin is a major component of the middle lamella, which helps cells to bind together. During fruit ripening, pectin is broken down by the enzyme pectinase. In the process, the fruit becomes softer as the middle lamellae are broken down and cells become separated from each other. Pectinase is commonly used to speed up the extraction of juice from fruit. Moreover, pectinase has been used in wine production since the 1960s. Pectinase helps the extraction of flavors. Also, the presence of pectin in finished wine or in fruit juice causes slight cloudiness. Pectinase is used to break it down, producing a clearer finished product. Because of this, pectinase is widely used in food industry. In particular, it is mostly produced by fungi such as Aspergillus niger.